Thank you. I'm glad it's quieted down. I'd like to welcome you all. My name is Susanne von Kemmerer, and I'm the Deputy Director of the ARC Center of Excellence for Translational Photosynthesis. And I'm also one of the organizers of this conference on C4 photosynthesis, which we've been holding in Canberra here. And we've organized this public lecture that you're going to hear in a moment. Now, before I begin with the proceedings, I would like to acknowledge and pay respect to the traditional owners of the land on which we meet, the Ngunnawal people of the Wiradjuri Nation. It's upon their ancestral lands that the Australian Academy is built. Now, I've also been asked to run through some general housekeeping before we start. In case of an emergency, your emergency exits are just next to those, further out those doors. The toilets are located on that side of the room. It's a real pleasure to welcome you here for this event. It's part of our meeting, as I've already said, celebrating 50 years of C4 photosynthesis and discovery and innovation. The talk by Rowan Sage will go for approximately 50 minutes, and then you'll be able to ask questions and answers. The event will also be recorded, and we hope to make the video available to you in a short time. May I ask you now, please, to ensure that all your mobile phones have been turned off so we're not interrupted. And I'm not sure, I was hoping to have the Twitter details on, but I don't. It was there? So yes, you can also tweet. Down there. For those of you who like Twitter. So, sorry. Can you go to the next slide, please? I seem to not be able to get the next button to work. Huh? And can you do the next one as well, please? Thanks. I would like to introduce our speaker for tonight, Professor Rowan Sage. Professor Rowan Sage is a professor of plant biology in the Ecology and Evolution Department at the University of Toronto. And Professor Sage's research addresses ecophysiology of C4 plant evolution. He's identified a number of independent origins of C4 photosynthesis and also is working on the drivers of evolution of C4 photosynthesis. His lab currently is identifying a number of species that are intermediate species and on the path to C4 evolution, and this will help to better understand how C4 photosynthesis has evolved. Welcome, Rowan. I'd like to ask you to come up now. Okay.
Okay. Is this one working? No.
Yes, but can you?
Yeah. Okay. All right. Well, thank you all for coming. I'm going to launch into it without much delay here because I have a bit of a story to tell you. I'd like to thank Hal Hatch and Roger Slack for discovering C4 photosynthesis and for a number of the folks here who helped contribute to that over the years. So I'm going to start not with C4, but with C3 photosynthesis, which was discovered earlier and is the most predominant form of photosynthesis on the planet. And as soon as I get my mouse working, there we go. Mouse. Whoop. Mouse doesn't work. There. It's another one of those rogue mice. Anyway, well. If I could get a pointer, I could point. The mouse is not working. Okay. Do you have a pointer? Yes. I move it and I don't see it. Can you? I can't see it here. All right. Well, so much for that. There we go. Okay, so the story begins with Rubisco here, the enzyme that takes CO2 from the atmosphere and binds it to ribulose bisphosphate, which then creates a 3-carbon molecule PGA. And using energy in the form of ATP and NADPH, you make sugars, which then go to feed the plant and build the plant in many different ways. and you regenerate the RUBP in what's called the photosynthetic carbon reduction cycle. And that's C3 photosynthesis, in short. Now, C3 photosynthesis is the first form of photosynthesis in eukaryotes and cyanobacteria on the planet. And it's about 3 billion years old. Currently, it accounts for about 75% of the terrestrial primary productivity on the planet, and it dominates in plants of cool environments. Now, critically important is all the trees on the planet, save for 2 or 3 species, are using this C3 photosynthesis, and that will become important later on. And the vast majority, over 90% of the food that we directly consume, is using C3 photosynthesis. So rice, wheat, cotton as shown here, all our fruit crops, most of our vegetables and fiber crops and so on. Now, C3 photosynthesis has a bit of a problem, and that arises from rubisco's ability to take oxygen instead of CO2, and it oxygenates ribulose bisphosphate, making this thing here, PG or phosphoglycolate, a 2-carbon molecule that's poisonous to the plant if it accumulates in high concentrations. And so the plant has evolved a mechanism using energy, again in the form of ATP and NADPH, to scavenge carbon out of this phosphoglycolate and turn it back into this PGA, which can then be made back to RUBP. But previously fixed CO2 is given off, and this leads to a wasteful reaction that reduces photosynthesis quite a bit. And we call this reaction cycle photorespiration, beginning with the oxygenation and then the release of CO2, just like with mitochondrial respiration, although by a different mechanism. Now this diagram shows CO2 in the chloroplast of a leaf and temperature, and the photorespiration to photosynthesis ratio. So basically where you have this high area here at low CO2 and warm temperatures, you have very high rates of photorespiratory inhibition. If you raise the CO2 levels, you inhibit the photorespiration reaction because CO2 outcompetes oxygen for the active site of rubisco. And then if you go to higher temperatures, that tends to stimulate the photorespiratory reaction by stimulating oxygenation. So if you get a combination of low CO2 and high temperatures, you get quite a bit of photorespiration. And these circles here indicate what would occur in recent atmospheres in a leaf as you went from cool to warm conditions. So in recent atmospheres, photorespiration could get to be quite significant in warm environments. And this diagram here shows net photosynthesis as a function of temperature. This is what you would see in a typical C3 leaf, a big, broad, looping response. And if you get rid of photorespiration, which you can do by reducing oxygen or raising CO2, then you see a much greater stimulation of photosynthesis by temperature. And this difference here between no photorespiration and with photorespiration represents the sizable inhibition you get. And at low temperatures, it's quite a small inhibition. At high temperatures, it's quite substantial. And so if you go into environments such as the tropical rainforest or a desert environment that you could have from the tropics to the warm temperate zone, photorespiration on a warm day can be quite substantial and lead to substantial inhibitions. Similarly, if you go to a rice field or a cassava field in the tropics, photorespiration is a significant limitation and cuts into yield quite a bit. Now, a mechanism was evolved, C4 photosynthesis, shown here, and instead of using the typical simple C3 cycle, it took the C3 cycle and packaged it into an inner tissue layer that surrounds the vascular bundles, and this is called the bundle sheath. And the bundle sheath you see here in an actual leaf cross-section is a big inflated layer of cells right around the vascular tissue shown there. And in here, rubisco is localized along with the rest of the reactions of the C3 metabolic cycle. And then out in this surrounding tissue, the mesophyll, you have upregulated a cycle starting with PEP carboxylase that will take CO2 and convert it into a 4-carbon molecule or a C4 acid hence the name C4 photosynthesis. And that's transported into the bundle sheath where it's decarboxylated to release CO2, and then it goes back, and using ATP energy, it regenerates an acceptor molecule that will bind to the CO2 and continue the cycle. And with each turn of the cycle, more CO2 gets pumped in here, and that raises the CO2 level in the leaf by perhaps 5 to 10 times over what it otherwise would be. And this specialized anatomy, which requires a bundle sheath and the mesophyll that's closely associated with it, we often call Cran's anatomy, to give it a name, after the German word wreath, which is what it sort of looks like. Now, C4 photosynthesis counts for about 23% of terrestrial primary productivity. It's mostly found in warm season grasses. There's only 2, maybe 3 tree species that do C4 photosynthesis. Our most productive plants, our world record holders for photosynthesis, our worst weeds are also C4, and then a handful of crops, such as maize, one of the top 3 crops in the world, sorghum, sugarcane, and millets are C4, as well as what are called the sour grasses here, which is a term that agronomists recognized before the discovery of C4. You had sweet grasses, which were cool season grasses of higher latitude, and then you had these warm season grasses they called sour grasses, because nutritionally they were not as strong for cattle and grazing animals at low latitude as the sweet grasses were in the cooler, higher latitudes. So if you go to a grassland such as you see here, the grasses in a warm climate would be C4, and all these woody species here would be C3. And so the tension between woodland and grassland is perhaps the greatest competition between the C3 and the C4 photosynthetic mechanism. And if we look at photosynthesis here as a function of temperature, here's that C3 response, which you saw earlier. The C4 advantage you see is like this, very similar to what you have if you remove photorespiration, with the exception that at low temperatures, the C4 advantage is lost because they require that extra ATP in part to drive photosynthesis. Anyway, but you see at warm temperatures, the C4s have quite a bit of a photosynthetic advantage, and that leads to them being more successful in warm climate regions of the world if it's competition between grasses, C3 and C4 grasses, for example. And so when we look at our bioenergy crops, which are heavily dependent upon grasses and herbaceous things, here's peak dry matter. The C4s, you see, producing quite a bit of dry matter compared to the C3s, and as a result of this, people are looking at using C4 photosynthesis to develop new crops for bioenergy, biomass, or specialty products production. And if we look at the biomes of the world, focus on the orange here. This is your C4-dominated grasslands. Notice they're mostly low latitude in northern Australia, in India, and so on, and in North America, in the southern states of Texas, Oklahoma, and so on. The green here is forests, and so wherever trees can establish, then the C3 photosynthetic system can dominate. So in the wet areas of Amazonia, you have tree dominance. In the dry areas of southeastern Brazil, the C4 grasses can be very important. So with this little bit of background, the question is where, when, and why did C4 photosynthesis evolve? And that will be the focus of today's talk. Now, people began to wonder and ask this question about where, when, and why C4 evolved almost immediately upon the 1966 publication of what we now call the C4 biochemical cycle. And John Downton in 1968 published a paper. He is an Aussie guy who was working with a Canadian guy for his postdoc, and he went and identified groups of grasses that had a low CO2 compensation point of photosynthesis, which meant more efficient photosynthesis, i.e. C4 photosynthesis. And he compared that with another group which had a high compensation point or were inefficient grasses. And now we know the low compensation point grasses clustered in this group here that we call the pack-mad grasses, which is one of the two major clades of grasses in the grass family. So we have one group here that's C4, another group here that's C3. Now another Aussie, or actually a Kiwi, John Troughton, went and quickly jumped on a discovery that came along in the late 1960s from a lab in Wisconsin that noticed that corn and other crops that are C4 had a lower different carbon isotope ratio. So what we have here is number of species as a function of the ratio of carbon-13 to carbon-12, and you have the lower values here, which are the C4 values, and you have the higher values here, which is where the C3 values clustered. And they noticed very early on that there was this very clear segregation in carbon isotope ratios between C3s and C4s. And using this technique, they quickly realized they could screen plants for either a C3 or a C4 photosynthetic pathway. And one of the things that John Troughton did is he went and looked at samples of coal from different geological ages, such as the Pliocene or the Permian, to see how far back in time C4 photosynthesis might have existed. And all the coal samples came back as being C3 in origin. But it shows that very quickly, within a few years of the first discovery of the C4 pathway, people were exploiting these new techniques to address evolutionary questions. And by 1970, they had already begun to map out the distribution of C4 photosynthesis on the tree of higher plants, the angiosperm tree here. And they identified it in a number of orders already. And in the next subsequent years, they identified it in these additional orders shown here with the green arrows. So by the time the early 1980s, we pretty much had a good idea of the distribution of C4 photosynthesis in the plant kingdom. Now, in 1974, an interesting discovery by Mac Leitch's group at Berkeley was that there were groups of plants that were intermediate between C3 and C4 plants, and we call these C3, C4 intermediates. And so in 1974, they identified one, and quickly in 1975, another one, a grass, Panicum mimoides, was identified. And in 1979, another one, and then in 1978 to 1981, another group of intermediates was identified. And as shown here, this is an intermediate photo. If you look at the bundle sheath cells, you see a lot of organelles in here, which is very reminiscent of what you get in Cran's anatomy. And we'll come back to these a little later in the talk. And they identified that here we have the response of photosynthesis to intercellular CO2 levels. Here's C3, and it has a relatively high CO2 compensation point of photosynthesis. It takes about 50 parts per million to have positive photosynthesis. The C4 has a higher rate at low CO2 and a very low CO2 compensation points. And the C3, C4 intermediates here have an intermediate performance in terms of photosynthesis. Now, in 1999, I was asked to edit a book that pulled together our understanding of C4 photosynthesis up to that point in time. And this is the result, C4 plant biology. And at the time, we identified about 25 independent lineages of C4 photosynthesis. They were all in advanced angiosperm orders. The thinking at that time was maybe C4 evolved 10 million years ago, but there was quite a bit of uncertainty. There were a number of these intermediate groups, but one of the biggest weaknesses, which was identified in a chapter in the book, was our phylogenetic understanding, or our understanding of the familial relationships between the different C4 plants was quite rudimentary. And here we have a phylogenetic tree put together based on DNA sequences for a number of proteins. And where you see bold, that would be a group that has C4. And this was compiled by Elizabeth Kellogg at the Missouri Botanical Gardens. And in this chapter, she did her best to pull together the phylogenetic distribution of C4 by mapping it onto these trees. You see here, for example, Anacampsaros is supposedly C4. Here's Cochia and another few supposedly C4s. But a big problem we have is there hadn't been much work, and so we didn't have good species coverage. So although we could place C4 at some point on the tree, we didn't necessarily know its relation with C3 species. And so we had very poor resolution of the C4 clades, which meant we had a poor ability to say this is exactly where C4 evolved. And so we couldn't use that then to go and ask questions about, say, what environmental conditions promoted C4, or were there traits that promoted C4, and so on. And so I took it upon myself, and I enlisted my colleague Tammy Sage from the University of Toronto, and we used John Troughton's isotope technique to screen herbarium specimens by taking a small piece of leaf sample and determining its carbon isotope ratio. And if it fell here, it was C4. If it fell in this cluster, it would be C3. And then we went and we did this for as many species within a group where C4 was known to occur in order to identify who was C4, who was C3, and then map it on a phylogeny. And to get the phylogeny, I didn't do it myself. I went and collaborated with many expert, excellent phylogenists, some of whom are in the room. Here's a number of them I've worked with. And we were able then to go and map C4 photosynthesis onto the phylogenetic tree. And so this is the Amaranthaceae tree. And you see all the different groups here branching off in their familial relationships. And then when we map C4 on, as we do here, we can find that there's 5 clusters. One here for Amaranthus, Urva here, a small cluster for Tetostromia, about 20 species for Alternanthera, and then about 80 species for this group, the Gomphronoids. And we can conclude that C4 independently evolved here, 1, 2, 3, 4, 5, because these clusters are separated by C3 species, and so we know that there's independent evolution. Now recently we've been continuing to do this, and recently we did it with the group Blepharis, shown here, and here we have a C4 cluster. Here we have a cluster that has C3, C4 intermediate traits, and here we have a cluster of C3 that branches in an ancestral position at the base of these branches. And so we know that from this, C3 is an ancestral state. These intermediates cluster in an intermediate position, and then the C4 is in a derived position within the tree. Now, with my colleagues and many other peoples, we were able then to map onto the entire angiosperm phylogeny, shown here as a circle diagram, the number of C4 origins. And where you see red, for example, up here with the grasses, you have a bunch of independent origins. Here in this Helianthae group, which includes Flavaria, you have one origin. And then in this group of many stress-tolerant plants called the Caryophyllales, you also have a cluster of origins. So you have 2 patterns. One is clustering with a lot of origins, and then a number of outliers that evolved C4 separately. Now, once we get this phylogenetic distribution, we can finally begin to go after addressing evolutionary questions. And with our understanding of who was C3 and who was C4, we can do an accounting for the number of C4 species on the planet. So we now know that there's 19 C4 families, or about 3% of all the plant families, 418 genera, and about 8,100 C4 species, or 3% of all species. This is made up of 5,044 grasses, 1,300 sedges, and about 1,800 dicot species. About half of the grasses are C4, a third of the sedges, less than 1% of the eudicots. And because there's so many eudicots, we come up with 3% of all plants. Now the life forms, again, there's very few trees, 2, maybe 3, maybe 4, I was liberal here and set up to 6. There's no climbing vines. There's 400 shrubs, mostly from the desert, 1,300 species of herbaceous plants, and 6,300 grasses and sedges. So these are a bunch of numbers, but the important thing is it's a small fraction of the total number of flowering plants on the planet. But this relatively small fraction of 3% of all plants accounts for 1/4 of the primary productivity on the planet. So these C4 guys are really carrying a heavy load with respect to contributing to the energy and biomass that circulates through the biosphere. Now, I want to come back to this Blespharis phylogeny. What you can do with these phylogenies is you can look at the sequence divergence between the various branches on a phylogenetic tree, and if you assume something about the rate of divergence, you can estimate when these branches occurred in geological time. So on this tree, it's been calibrated. Here's the 10 million year point. Here's the 20 million year point. And the Blespharis C4 origin here is estimated to have occurred approximately 5 million years ago. And my colleague Pascal Christian and a couple of us, he's here in the room, he went and dated the origin of C4 for a large number of lineages, approximately 48 lineages here. And then he plotted it against geological time from the Oligocene to the Miocene to the Pliocene, or over the past 30 million years. And then we linked that up with changes in CO2, changes in the appearance of fossils in ecosystems, and then this parameter, change in oxygen isotope ratio, which tells us something about the climate. And as you can see here, the C4s all originate in the last 30 million years at a time which corresponds to a drop in atmospheric CO2 from fairly high levels before 30 million years ago to levels that are quite similar to today over the past 25 million years. Now, I took these midpoint values here in the phylogenetic tree. These are the midpoints between what we call the crown branch node and the basal branch node. And so I just took these midpoints to set up a little simpler graph. And here we have the midpoint estimate of age for all the C4 lineages that we can do. And I took the species numbers for each of these lineages and plotted species per lineage against age. And what you see here is the older lineages, such as the chlorodoid grasses, the andropogonoid grasses, which includes maize and sorghum, euphorbia, which is a dicot group. You see that the speciose groups tend to be the older groups. But the younger groups, less than 8 million years, all have very small numbers of species and very low diversity in terms of form and geographical distribution. And so this is a sign that the older groups are the ones that have speciated and are the most important in terms of, say, grassland dominance and producing net primary productivity. And then I also put the numbers of origins into these bins of 5 million years, and you see there's a few that start early on, and that would be these oldest speciated lineages, and then you have a number that come in more recently. And this becomes important because we can now link this with changes in CO2 and climate. Here we have a more recent CO2 record put out by a guy named Mark Pagani. And you see the CO2 is quite high before 35 million years ago. This is the CO2 that we might have in 100 years if we don't get global warming under control. And then the CO2 drops down between 30 and 25 million years ago, is about 400 parts per million to 450 parts per million, and then it drops down at about 14 million years ago to levels that were similar to what we had in the pre-industrial period, and then in the Pleistocene, the ice ages, it drops even further. Now, when we put on the CO2, or the number of lineages and their appearance, you see it starts as the CO2 is coming down, and then it really takes off right after this drop here, and that implicates CO2 as an important driver for C4 evolution. Now, the other thing we can do is use this to identify and place these intermediate clades, and going through many of the other orders, we've been able to identify over a dozen clades or lineages that have C3, C4 intermediates in a close, or what we call a sister position to the C4 clade. And then we can use these to address questions of how C4 evolved by looking at the characteristics in these clades and using it to address questions of how C4 might have evolved. The other thing we can do with this phylogenetic understanding is we can go and look at the species that cluster around the branch points where C4 arises. We can also look at where the intermediates occur in the clade, and where you get a combination of intermediates and closely related C3 and C4s, we call that the center of diversity, where C4 probably evolved. And if we put up the center of diversity for the many lineages that we've been able to identify, you see that they cluster in these sort of warm subtropical dry areas, such as the Chihuahuan desert of North America, the dry areas of southeastern Brazil and Argentina, southern and southwestern Africa, northeastern Africa, here in the Arabian Desert and the eastern Sahara, dry Central Asia, and then 4 lineages in Australia, mostly Western Australia, out in the dry, arid areas. And this allows us then to ask, address hypotheses about the environmental drivers favoring C4 evolution. And you can go to these sites and actually collect things like euphorbia intermediates, such as this guy here, or heliotropium intermediates, such as this guy here. There's a close-up of him. Or in the Borhavia clade, you can go here and collect intermediate species, and then look at the environment. And when we do this, here's one of the Australian intermediates right here, growing in this this rocky pothole that's formed in a very harsh condition out in Western Australia. And when we go and look at all the intermediates and compare their environments, we find that they consistently grow in areas with very high summer temperatures. And how high is shown here. We measured the temperature on these guys, Heliotropium convolvulaceum, an intermediate in the Heliotropium lineage. And we looked at summer temperature, and it starts out in the mid-20s, but by noon it gets up close to 50 Celsius. And in low CO2 of recent times, this high of a temperature would allow for very high rates of photorespiration. And so we also find they grow in semi-arid to arid climates consistently. They grow in sand, gypsum, rocky, or saline soils, such as these sandy things. These are very hot surfaces. You burn your feet if you walk on this. Temperatures get up above 60 Celsius. They often grow where there's very low competition due to extreme soil and disturbance. But you do get episodic summer rain, which is important because it allows for plant growth in a very harsh condition. If you had no rain, you just wouldn't have any plants there. Okay, and so we call these conditions, these hot conditions, often on altered soil or disturbed landscapes with dry, sometimes salty conditions. We call this the nursery for C4 evolution because it leads to extreme rates of photorespiration, which on the one hand drag C3 photosynthesis down, and on the other hand promote the possibility for evolving alternative physiologies that might deal with this high level of photorespiration. And so the question, how could photorespiration promote C4 evolution? So here we have a diagram of the photorespiratory biochemical cycle. Rubisco takes in oxygen, produces phosphoglycolate, which is then converted to glycine in an organelle called the peroxisome. And the glycine goes to mitochondria, where an enzyme known as GDC, or glycine decarboxylase, will take the glycine, convert it to CO2 and serine. The serine will be made back to PGA in the photorespiratory cycle. and the CO2 was always sort of thought to have escaped. Now, under high rates of photorespiration, this CO2 here becomes a valuable resource for the plant. It's CO2 that's already in the plant. So if the plant can somehow compartmentalize the release of this photorespired CO2, it has a means to turn it, instead of a waste product that can escape, into a valuable resource that can enhance photosynthetic efficiency. And so this is how they've done it. What they did is they created a photorespiratory cycle that has to run over 2 cells. So in the mesophyll cell, you make glycine, and you knock out GDC expression in mesophyll mitochondria. This forces the glycine to diffuse into another cell, which in this case is the bundle sheath cells, and here you have a lot of mitochondria with the GDC expressed. And then those mitochondria will take the glycine that flows in from the mesophyll, convert it to CO2 and serine, and then the CO2 can be taken up. And here's the same thing, just another version of it. Glycine goes in, mitochondria with GDC react it, creating CO2, and serine, the serine goes back. And so we call this the photorespiratory 2-celled cycle, or to be simple, we call it the C2 cycle. And so this then is a way that plants can scavenge and recover photorespired CO2 and enhance their fitness. This basically is the C3, C4 intermediate physiology. Now, once this happens, it's been hypothesized that you can upregulate what we call the C4 metabolic cycle, where CO2 comes in, bicarbonate is converted to bicarbonate, and then PEP carboxylase, the enzyme in C4 photosynthesis, will take PEP and create a 4-carbon molecule, which can go into the bundle sheath where a decarboxylating reaction can release CO2, which can be refixed, and then glutamate, ammonium here from photorespiration, can be tagged to the carbon skeleton, creating glutamate that can then return and be recycled. And it's thought that the upregulation initially of this C4 cycle is to create carbon skeletons to recover this ammonium. But because it's releasing this CO2, then it becomes adaptive and improves photosynthesis for the plant, and hence natural selection would favor more and more of this, eventually leading to C4 photosynthesis. And just to show a picture in how this looks like, here we have our friend Heliotropium convolvulaceum. Here's the mesophyll cells and the bundle sheath cells. And we're going to look at the electron micrograph of this bundle sheath cell here. This brown shows an antibody that binds just to GDC, and in the brown location of the antibody is just right along this inner layer of the bundle sheath where the mitochondria with GDC are located. And here we look at a bundle sheath cell, this cell here, and what you see is a lot of chloroplasts, and lining the wall of the cell underneath the chloroplast is a big layer of mitochondria right there. Here's another image. And this is what we see happening in these C3, C4 intermediate species. And so it's a mechanism then to scavenge high amounts of photorespired CO2. And so people have built models, beginning with Russ Monson, and this is my version of the model. And so the idea is that these ancestral C3 plants growing in these hot areas with stress, drought, will make a lot of photorespired CO2, and then you'll get the evolution of C2 photosynthesis, or this photorespiratory scavenging mechanism to recover the photorespired CO2, and then you enhance PEP carboxylase and a C4 metabolic cycle and strengthen it, optimize it, and then you end up with C4 photosynthesis. And there's been some question then, what happens to go from the ancestral C3 condition to this photorespired CO2 condition? And doing some brainstorming some years ago, I proposed initially that these arid zone plants, what they do to survive in these very hot conditions is, one of the first things is to enhance vein density, as shown by this simple little cartoon here. And this is sort of like what you would have in a very dry environment, you're going to have very high transpiration rates, very high rates of water loss through evaporation from the leaf. And to supply the water to all the cells in here, you just increase the number of highways or the number of road networks, that is, the veins, and they will bring the water in at a faster rate to keep up with the rate at which it is lost. And when you look in to the literature at the vein density of desert plants from hot climates, you find that they do indeed have a considerably higher density than those from cooler or wetter climates. And then there has been some speculation by a number of us that you also can inflate the bundle sheaths, perhaps as a way of increasing water storage and hydraulic capacitance so that you can supply water during what we call transpirational surges, where you might have wind, say, that all of a sudden increases transpiration, and then this storage area here can supply the water to the nearby photosynthetic cells. And when you get the bigger vein, vascular bundle sheaths here, then you may as well put chloroplasts and mitochondria in them and activate their ability to do photosynthesis. And Pascal Christian came along and did a very nice study showing that in the grasses, at least, you do have a wide, big veins in many of the sister groups to the C4 things. And also, if you look at poster number 34 in the meeting, you'll see a discussion of the potential significance of big veins in, and their role in promoting C4 evolution. So this just shows an image then of a big vein in a C3 relative to a C3, C4 intermediate. And this is quite commonly seen in many of the grasses where the C3 relatives, for some reason perhaps as a drought adaptation, have these big bundle sheaths surrounding the veins, and that could enhance the hydraulic integrity of the leaf. So this is the model as we're now working on it. You start out with anatomical preconditioning in dry environments, such as having close vein spacings or just a lot of veins. You then enlarge the bundle sheath cells, and you put in more organelles like chloroplasts and mitochondria in there. The mitochondria, for a reason we don't understand, become polar in their distribution. They move to the inner bundle sheath wall, and we call this the protocrans condition. And from here, you move to the photorespiratory CO2 pump by losing glycine decarboxylase expression in the mesophyll and only expressing it in the bundle sheath. So that leads to the photorespiratory bridge to C4 photosynthesis. So on the one hand, photorespiration acts as a drag on C3 photosynthesis, and then alternatively, it can provide the resources that natural selection can work on to build the C4 pathway. So I can stop here. I can go into the really cool part of the talk for 5 minutes. Okay. So the cool part of the talk is, what does all of this have to do with the fact that you are sitting here in this room right now? Could the evolution of C4 photosynthesis have led to the evolution of Homo sapiens? And this is coming off of some really cool work that's ongoing at the moment with a bunch of anthropologists who work on carbon isotope ratios and early hominid populations. And this is sort of the logic behind all this. We're declining CO2, which increases photorespiration and aridification, leads to C4 plant evolution, as we've discussed. This leads to the spread of grasslands. It reduces the performance of C3 plants, which at low latitudes is mostly trees, and this opens up the landscape, creating mixed C3, C4 communities, and it's on these open landscapes of C4 grasslands and partially wooded savannas that the hominins evolved. And here's a summary of that. Here we have the hominin branch beginning to diverge from Australopithecines and going up to Homo in East and Southern Africa about 3 to 4 million years ago. And this is a time when we see an increase in C4 biomass, as shown here with Slale carbon and isotope ratios from C3-like values at 4 million years ago to C4-like values as we come towards 3 million and 2 million years ago. And then here's the increase in grazers, which only eat grasslands. So the grasslands are opening up at a time that's coincident with the divergence of hominids. This is the divergence of hominids in another paper, and here's a number of the traits that make us human and when they evolved. Bipedalism came up about 3.5 million years ago. C4 consumption is apparent based on isotope records. Australopithecus is here eating foods of a mixed nature that gives it an isotope value halfway between grazers on C4 and browsers on C3. So our ancestors were feeding on something that originated as a C4 source. We began to eat meat. There's evidence of butchery here at 3.5 million years ago. And by 2.5 million years ago, you see evidence of stone tools and something. Now, here's some more evidence of that. As you go to more open grassland, shown here by the lighter symbols, the isotopic ratios shift towards C4 values in various hominid groups, such as the Australopithecines here and later on Homos. Now, this is the landscape that it would have looked like, and this shows sort of the role that CO2 plays in all of this. And this we're seeing today. So at current CO2 levels that are high in this landscape, you see a high density of trees and shrubs. This landscape is becoming wooded. If you go to low CO2, you see it's very open because the trees and shrubs are not doing so well under low CO2. And this is an experiment done by Bill Bond. He grew acacia trees at low CO2. This is the CO2 levels of the Pleistocene. And you see the roots are quite scrawny. And at higher CO2, the roots are very robust, which would make these plants these trees able to establish in a grassland fairly effectively. This is what the grassland would have looked like. And what I want all of you to do now is imagine yourself bare naked with nothing but your hands and maybe a few rocks and sticks running around on this landscape. And these are some of the guys that you might be interested in eating. And these guys are important because this biomass here of C4 grasses, we can't really eat it very effectively. We don't have the digestive system to digest it. The seeds shatter easily and scatter about, so it's not efficient to pick them up. But if we can harvest these guys who can effectively convert this C4 biomass to protein, this very abundant C4 biomass, then all of a sudden this open landscape becomes a very effective niche, a very productive niche. And so all we have to do is figure out how to get this version of C4 biomass into our bellies because we can't eat this version of C4 biomass. But there's one other guy out there we have to worry about, and if you're running around bare naked with just some sticks and rocks, a big lion like this, who's 2 to 3 times your size, will obviously have you for dinner. And so to live out on these open landscapes, to exploit this biomass here, the animals, we have to be able to watch out for these guys. And so that creates a very strong selection environment for the traits that make us human. And so using one of our well-known C4 biologists here, who's now working on things like C4 engineering into crops, a number of features here. Bipedalism, that allows us to stand up above the grasses and see out over the grasses so we can see the foods and see the predators. Also, upright stature. An important thing we can do is we sweat. Most animals don't sweat. Now that becomes important because what it allows us to do is go out into the hot middle part of the day to hunt and scavenge. That's when the lions and tigers and bears or whatever are going to be taking a siesta because they cannot go out in the hot part of the day because they cannot dump the heat. So sweating opens up a midday niche where we can actually live on the grasslands. And our ancestors, actually the chimpanzees and the apes, are not very good at sweating, nor are the predators. Distance running, that evolved at about the same time, allowing us to chase down the prey or run over wide landscapes. Teamwork, this became important because on the open landscapes, you all by yourself, bare naked, are not going to last very long. But if you're with other people, then you have people watching, you have people potentially defending. A lion may not come up against a bunch of you screaming and throwing rocks at him and so on. And that leads to coordinated defense. Also, extended child care becomes very critical because to have big brains and to be smart enough to live out there, it takes a long time for the babies to grow up into adults, and so you have to have a coordinated network of family care to raise the children. And all of this takes increased brain size. And so the C4 grasslands, which spread because of C4 photosynthesis, created the niche for our species to go out there and be selected for and evolve into who we are. So with that, C4 plants are important for a lot of reasons. If for nothing else, then if they never evolved, none of us would probably be here. So thank you.
Thank you very much for a very exciting talk. Rowan has agreed to take questions. So if you're interested in asking questions, please raise your hand and someone with a microphone will come to you. Is there any? Yeah, there? Just wait.
Yeah, I guess I read about this, but just I wonder whether you can elaborate what are the geological events driving the drop down or gradually dropping down of the CO2 then?
It's a number of things, but a few of the big ones is India smashing into Asia and causing the uplift of the Himalayas. So when you get more exposure of rock, young rock, erodible rock, to the atmosphere and to water, as that rock mineralizes, it grabs the CO2, and then the CO2 comes down as a carbonate and gets deposited in the ocean as limestone. So when you get a lot of mountain building, you get a lot of sites for CO2 sequestration out of the atmosphere, and so that helps pull the atmosphere CO2 levels down. Other things then, as it gets colder, and then you can begin to form ice at the high and low parts of the planet, and that then acts to cool the planet, cool the oceans, and a cool ocean is a bigger sink for CO2, so that feeds into this as well. And a wild thought is C4 grassland expansion changes the surface albedo of the planet a bit, and that also will then act to cool the planet down quite a bit. Albedo is very important, and to give you an example, at the end of the last ice age, the CO2 change from 180 ppm to 260 ppm accounted for about half of the warming of the planet. The other half came from changes in the albedo when that white ice cap at the top of the Earth and on the mountains and so on was replaced with vegetation. So the boreal forest and that albedo shift accounted for about the other half of the warming at the end of the ice age. So vegetation strongly interacts, and we could think that the spread of the C4 grasses 10 million years ago to 5 million years ago helped push the planet into the deep ice age that began in the last 2 to 3 million years. I don't know, Colin, what do you think of that idea? You follow this literature too.
Are there other questions in the audience? Yes, over there. Just wait a moment.
Thanks, Rowan. I guess maybe just to ask the question that maybe others are thinking, what is the future for C4 photosynthesis as you start describing this increase in carbon dioxide in the atmosphere, especially in the natural ecosystems?
Unfortunately, as CO2 goes up, CO2 is not bad for C4 plants directly. They grow a little bit better, their water use efficiency goes up, but it's really effective for the woody species, the C3s, and so they can encroach and spread into the grasslands and convert the grasslands to woodlands. And we're seeing this happening across the planet at the current moment. Now, certainly land use change like overgrazing also helps suppress grasslands, but the coupled feature of overgrazing and land abuse with higher CO2 is leading to a large amount of C4 grasslands being displaced. So the habitat for C4s are going to shrink, and with that we're going to probably lose a good chunk of the C4 flora. So when a few of you who are very young now might be here in 50 years ago meeting on the 100th anniversary, and hopefully we'll have solved the CO2 greenhouse gas problem and we'll manage our resources so there will be a very robust C4 flora out there. But at the way things are going, there's a good chance that instead of raising a toast to celebrate the C4 flora, they might be raising their glasses in a long lament for what's been lost. So it's pretty dire for a lot of those species. Certain ones will do fine, like the really weedy ones, and the crop ones that humans need, they'll do fine, but ironically, some of those weedy ones will get rid of much of the natural diversity because the weedy ones respond so well to human land use practices and often human abuse like overgrazing. So.
Thank you.
Susanna, we have a question up top.
Oh, that's good. Do we have a microphone up top?
Yeah, I got it here.
Thank you.
So we have archaeological evidence of early humans. Do we have any evidence of C4 plants gaining dominance that would coincide in terms of timing and geography with these findings of early humans?
Yeah, I showed you a sort of a simple summary of that here. So these are three pieces of evidence from the geological record. This one is carbon isotope ratios of plant waxes that have been deposited on the ocean floor just off of East Africa, close to the Turkana Basin, which is where this hominid fossil assemblage was identified. And it's going from -28, which is somewhat of a C3 value, and drifting to -24. And the leading explanation for that is a mixture of C4 grasses coming in and depositing their plant waxes along with a bunch of C3 trees. Here we have carbonates, and these are fossilized carbonates that are deposited by carbon coming out of the decomposing organic matter that settles into the soil. And the carbonates here have a C3 isotopic signature, and then over the 3 to 4 million period, it shifts more to a less negative value, which is indicative of C4. And then this doesn't show the carbon isotopes of the animals, but if you take the tooth enamel of the animal, you can estimate what it's eating. And there's a large amount of evidence that shows animal tooth enamels are shifting from C3 to C4, beginning in about 10 million years ago and occurring through to this period here of about 2 to 3 million years ago. So yeah, there's quite a bit of isotopic evidence that couples with things like the appearance of C4 vegetation in the human diet, which is based on carbon isotope ratios in the tooth enamel. So, and these references, if you get this reference here, if you want to write it down, that's a recent review that summarizes the best of the latest data. It's quite comprehensive.
Thank you, Rowan. I think we take one last question over there.
Thank you, Rowan. I had a question. I saw that the evolution of the C4 lineages has slowed down in the last 3-5 million years, whereas CO2 has continued to stay low. I was wondering whether you have a clue about that.
You're gonna have to ask me that one in a few years, because as we get into the really young lineages, I think to a certain extent the phylogenetic resolution goes down because you no longer have quite the same stem and crown node separation. Oftentimes you just have the stem node, and I think as we get more phylogenetic resolution, we can pinpoint those times, and then I can give you a better answer as to whether that's real or whether it's a phylogenetic artifact. I have to emphasize that those estimates of lineages are hypotheses, and as we get more data, we can begin to refine them much more and narrow those uncertainty bars that would exist around them and come up with much more robust hypotheses. And at the time of the 100th C4 meeting in 50 years, I think that those timelines will be fairly well settled by that point. That should be one of the crowning achievements of many of you scientists who are in the room now, you youngsters who will continue this research. Okay?
Thank you, Rowan. I think perhaps I can ask you all to thank Rowan one more time for a very interesting talk. So this brings us to the end of the proceedings, and I thank you all for coming along, and I wish you a good evening. Could I ask the participants of the conference to stay behind? We're going to try and take a photo, and perhaps also the people that are up in the balcony and who are participating in the conference of C4 photosynthesis, if they could come downstairs. Thank you very much.